Okay, thanks Natalie. So I'm going to present the work that's been done by our PhD student, Hayley Jorgensen. There's a picture of her there. And it's about the impact of offshore wind farms on benthic microbial communities. So when we think about biodiversity, we think about these sort of enigmatic species like the birds and the fish. And what kind of gets overlooked are the microorganisms. And as we've heard earlier in Ben's talk, these microorganisms are of fundamental importance. For example, they drive some of the macronutrient cycles that we see in pretty much every environment. So things like nitrogen-carbon cycling, these are really, really crucial in marine systems. And you've got things like ammonia-oxidizing bacteria and archaea, nitrifying bacteria and denitrifying bacteria in the water column, and similar organisms found in the benthic sediments. And these organisms represent a reservoir of genetic diversity and metabolic potential. But the question is, do these man-made structures affect these microbiomes, and in turn, does that affect the biogeochemical cycling of nutrients like nitrogen? So this is a really complicated picture, and I'm going to try and walk you through it. But this is basically the nitrogen cycle, and really it's just split into two parts. There's the oxic processes over here on the right, and then the anoxic processes on the left. And one of the main processes occurring in the oxic regions is nitrification. And that involves the oxidation of ammonium to this intermediate hydroxylamine, nitrite, and nitrate. And a key critical step of nitrification is ammonia oxidation, shown here. So this initial step, this oxidation of ammonia to hydroxylamine, involves two key functional groups of organisms called ammonia-oxidizing bacteria, or AOB, and ammonia-oxidizing archaea, AOA. And these two groups of organisms have an enzyme called ammonia monooxygenase, and that catalyzes that bit of the reaction. And there's a gene that we can use as a target that encodes for that enzyme called the AMOA gene. So we can use this as a kind of proxy for looking at nitrification. So the nitrate that's produced from nitrification can then get reduced in a stepwise manner in the anoxic zones by denitrification. And you can see there's lots of different enzymes that are involved in denitrification, and each one of these are encoded by different genes. So, for example, NAR and NAP, NERS, NERK, and NOS-Z. And I want to just flag to you about NOS-Z, and I want you to sort of remember about NOS-Z. So NOS-Z is pretty interesting, and this encodes this last step of denitrification. So if we're getting this last step occurring, then we get complete denitrification to dinitrogen gas. If this doesn't happen, we get partial denitrification, and then you get the production of greenhouse gases like nitrous oxide, which obviously we don't want to have happen. And of course, denitrification leads to a net loss of N from the ecosystem. So in healthy sort of systems, the nitrification-denitrification processes are tightly coupled. So there's a few gaps that we are still needing to do some more research in, and this is where Hayley's studentship is trying to address some of these gaps. So one of the questions is, how do benthic microbial communities change during the lifespan of a man-made structure? And Ben gave some really nice data on that for the Hutton and Miller sites. And then how do man-made structures alter the sediment physicochemistry, which in turn alters these microbial-driven nitrogen cycle processes? And are there any other environmental factors that we need to be considering that as a result of these man-made structures that impact on this microbial function? So, as you're probably aware, some of the sort of common environmental monitoring surveys include things like fisheries, ornithology, etc., and all these surveys here, among others. But what is missing is the microbial biodiversity, and in particular, the relationship between that biodiversity and the biogeochemical nutrient cycling. And this is unbelievable that this is being missed, given the fundamental importance these processes are, in particular to marine health. So, can we use molecular techniques, eDNA techniques, to identify particular taxa or gene proxies that we can maybe use as biomarkers that can then be added to these environmental monitoring surveys? So one of the main aims of Hayley's PhD is to characterise changes in the abundance structure and function of these microbial communities in relation to these structures. And we hypothesised that compositionally and functionally distinct microbes would exist in close proximity to these structures that may be more resilient to contaminants, for example, hydrocarbons and metals, compared to those communities found further away. So this studentship involves two different types of structures. There's the oil and gas structures, and you've already heard about the Northwest Hutton and Miller. So I'm not going to talk about that today, but what I am going to talk about is the other structure that Hayley's focusing on, which is the wind farms. And this at the bottom shows you the range of molecular techniques that we are using for both of these types of structures. So we're doing things like qPCR, which is a quantitative measure of abundance, microbial abundance, so asking the question, how many are there? And we can do this targeting different genes, so taxonomic genes and functional genes. We can sequence them to get the question of who's there, so the diversity, and metagenomic sequencing to get the question of what are you doing, the function. And we can relate this information to some of the physicochemical parameters and then plug those into some models. So you've already heard from Natalie the sampling location that we did of the offshore wind farm. It was here at Barrow, which is off the coast of Morecambe Bay. And this wind farm was operational in July 2006. And this was the schematic you've already seen of our sampling location. But just as a reminder, for the purposes of the data that I'm going to present to you, we've got some samples that were taken in the centre of the wind farm that we're calling 0 metres. And then we've got samples at 50 metres, and then what we're calling our control site at 3,200 metres away. So we're considering that as the sort of less impacted site. And these sediments were sectioned into the upper oxic layer, which we're calling 0 centimetres, and a deeper anoxic layer at 5 centimetre depths. And what I'm showing you here is some of our qPCR data. So this is our abundance data, the how many are there question. And what we can see is this is the 3 domains of life, the bacteria, the archaea, and the fungi. On the left-hand panel is our top surface layer, our 0 centimetre, so the oxic layer. And on the right-hand side here is the deeper 5 centimetres, the anoxic layer. And what was quite interesting here at the 3,200 metre site, so our control site that's away from the wind farm, we're seeing a dip in the abundance across the 3 domains of life that is only happening here at the 0 centimetre depth sediments, and pretty much similar abundances seen in the deeper sediments. So that's the sort of general community abundance. Now let's have a look at what's happening with the functional communities, and in particular those involved in the nitrogen cycle. So you remember these 2 groups of organisms, the AOA, the ammonia-oxidizing archaea, and the AOB, ammonia-oxidizing bacteria. These are the nitrifiers, so these oxidize the ammonia to hydroxylamine and then nitrite. So here we've got the AOA, here on the left, and here on the right we've got the AOB. Top slice, the 0 centimetres at the top here, and the deeper 5 centimetres here. And the first thing to sort of note is that there's a greater abundance of AOB to AOA, sort of a couple of orders of magnitude more AOB than AOA, which suggests that the nitrification processes that are occurring at this location are probably being driven mainly by the AOB. But also interestingly, again, in the 0 centimetres here with the AOB, we're seeing a dip in abundance of AOB at the 3,200 metre site that we're not seeing in the centre of the wind farm, and we're not seeing with the AOA, and it is pretty consistent with the deeper sediments at the 5 centimetre. And now if we have a look at the denitrifier genes, so we've got here the NERS and NERK, and I told you to remember the NOS-Z. This is the NOS-Z here at the end, so this is the one that determines whether we get complete denitrification or whether we get the production of nitrous oxide. And you can see here, this top row is the 0 centimetre, the oxic layer of sediment, and at the bottom here is the 5 centimetre, the deeper sediment. And again, we're seeing a dip here across these 3 gene targets at the control site. So basically, irrespective of what gene we have been targeting, we are seeing greater abundances of these microbes, including these key functional groups of microbes, closer to the structure compared to when you move further away. And so the question is, why is that? And are the particular taxa being stimulated by these wind farms? And so we want to try and have a look at what the shifts in the community diversity are, so the who's there question. And we're just in the process of doing some sequence analysis of the taxonomic and functional genes. We don't have that data yet, as well as some metagenome sequence data. So hopefully that will address that question down the line. And what we also want to do is to try and see if there are any environmental parameters close to the structure that might be driving these shifts. So for example, is there elevated nutrient levels? Are there some contaminants there, maybe some metals that could be either stimulating or causing some resilience to some of these communities? So we're thinking about metals, and we want to do some metal analysis of these sites, of these samples. But what I'm showing you here is a table that was pulled off from this report by Bowind, which showed some of the metal data from Barrow taken before it was in operation around 2002, 2004, and then a year after operation. And really what I just want to flag to you is some of these metal concentrations. They're all sitting within the OSPAR ranges. But one of the things that we're kind of thinking about is some of these metals, like copper and zinc, for example, could be micronutrients that these microbes can use. Copper in particular is used by the ammonium monooxygenase enzyme. As you remember, I said that is involved in the ammonia oxidation step of nitrification. So could this be something that's causing this stimulation of these communities? Alternatively, there could be some toxic metals maybe present, like chromium or lead, and maybe these communities are being resilient to that. And so that's something that we're going to look at going forward. And one of the things that Hayley is doing to try and address some of these questions is she set up this huge, massive FuECoMMS experiment, and it took like over the whole lab when she was doing this. It involved thousands of bottles. This is just a snapshot of a subset of those bottles. But what she did was she took some samples of sediment from the centre of the wind farm and at our control site at 3,200 metres away, and she incubated these sediments over time with a selection of metals shown here, and these were incubated either as individual metals or as pairwise or in 3s and 4 mixtures over this range of concentrations. And then we're going to do some similar eDNA approaches on this FuECoMMS experiment, as I've just started to describe to you for the field data. And we'll hopefully watch this space with interest and be able to report back on that in due course. So Really, I just want to sort of fly the flag for microbes really here and remind everybody that when we're thinking about biodiversity, we need to include the microorganisms. These are fundamental to the ecosystem functioning of these marine systems. They drive important nutrient cycling like nitrogen, also carbon as well. We saw from Ben's talk that they also play a role in sulfur cycling. And so these should not be ignored when we're thinking about the environmental monitoring surveys of these structures. And how can we do this? How can we include this? Well, eDNA is one approach that we can do, and Ben very eloquently explained to us the advantages of eDNA. So this provides us with a relatively rapid and high throughput analysis, and we can target different genes, we can target different taxa, and so we can get a lot of data from a very small amount of sample. But the question still remains is which taxa and also which genes should we be targeting as biomarkers? And we're hoping that we should get a better handle on this as some of this data comes through from Hayley's PhD. And I think that's it. And just want to thank all the guys shown here who were involved in the collection of the samples from Barrow. And thank you guys for listening.